Too often, genetic disorders result in the cell-by-cell decimation of particular tissues, muscles in DMD, nerve cells in Huntington and Alzheimer. There's no quick fix to this kind of insidious decay. But though these are early days yet, I think there is a realistic chance that we will eventually be able to treat diseases like these using stem cells. Most cells in the body are capable only of reproducing themselves. A liver cell, for instance, produces only liver cells. But stem cells can generate a variety of specialized cell types. In the simplest case, a newly fertilized egg, the stem cell with maximum potential, will ultimately give rise to every one of the 216 recognized human cell types. Stem cells are accordingly most readily derived from embryos. They can also be found in adults, but such cells tend to lack that embryonic ability to differentiate into any cell type. We are beginning to learn how to induce stem cells to produce particular cell types, and someday I hope we will be able to replace the lost brain cells in people with Huntington and Alzheimer with new healthy cells. But I caution that we have a long way to go before we have a thorough understanding of the molecular triggers that cause a cell to develop in one direction rather than another. It will take 10 years or so of grappling with this fundamental problem in developmental biology before we are in a position to explore properly the therapeutic value of stem cells. I think it would be a tragedy for science and for all people who may eventually benefit from stem cell therapy if research is hindered by religious considerations. Polls consistently show that the majority of Americans favor research using embryonic stem cells. And yet, politicians continue to pander to the outspoken religious minority that opposes it. The result is restrictive legislation in the United States that is hampering efforts to develop this potentially valuable technology. For now, treating genetic disorders does not extend to the wholesale replacement of cells, a la stem cell therapy. But it may involve replacement of a missing protein. Striking 1 in 40,000 individuals, Gaucher disease is a rare condition, resulting from a mutation in the gene for glucose cerebrosidase, an enzyme that helps break down a particular kind of fat molecule, which otherwise accumulates harmfully in the body's cells. The disorder can be devastating with a suite of symptoms, including bone pain and anemia. Initial attempts to supply the missing enzyme directly were made as early as 1974. The results were promising, but the logistics were nightmarish. The replacement enzyme had to be extracted from human placentas, and 20,000 placentas were needed to furnish a year's supply for a single patient. A big breakthrough came in the early 1990s when researchers synthesized a modified form of the enzyme, one that was taken up more efficiently by the cells that most needed it. In 1994, Genzyme, a biotech company, started to produce the modified form using recombinant methods. Treatment of Gaucher does not combat the genetic root of the disorder, but rather the effect of the mutation. It provides the patient with the vital protein that the faulty gene cannot. Righting the genetic abnormality via this biochemical route is evidently feasible and effective. But even given the remarkable efficiency of recombinant methods, the treatment is expensive, $175,000 per year. And the need for continual infusions imposes a burden on patients. Naturally, therefore, geneticists have long dreamed of a practical way to fix the cause of the problem rather than compensate for its effects. The ideal treatment for genetic disorders would be a form of genetic alteration, a correction of the genes that cause the problem. And the benefit of such gene therapy would last the patient's whole life. Once fixed, it's fixed for good. There are, at least in principle, two approaches. somatic gene therapy, by which we change the genes within a patient's body cells, or germline therapy, by which we alter the genes in a patient's sperm or egg cells, preventing the transmission of the harmful mutation to the next generation. Such solutions to the ravages of genetic defects might be obvious, but the idea of gene therapy has not met with the warmest of professional or public receptions. Such reactions are not altogether surprising. A culture wary of genetic modification in a corn plant might be expected to be averse to transgenic people, GM humans, if you prefer, however great the potential benefit. And more vociferous objections are made, also not unexpectedly, to the germline approach because of the risk of causing genetic damage when manipulating the DNA. In somatic gene therapy, such damage may be limited in its effect. In germline therapy, the possibility exists of accidentally producing impaired people. Even its proponents, of whom I count myself one, would never suggest that such a procedure should be carried out until our techniques are good enough for us to be confident that we will not inadvertently cause damage. Many scientists are convinced, though, that we should never attempt germline gene therapy. Whether based in ethics or unfounded fears of the unknown, such arguments are ultimately not compelling in my judgment. Germline therapy is in principle simply putting right what chance has put horribly wrong. But for now the controversy is academic. Germline therapy is still way beyond our technical powers. Until it is in reach, we should concentrate our efforts in making somatic gene therapy a powerful tool in its own right. The first apparently successful gene therapy was carried out by French Anderson, Michael Blais, and Ken Culver at the National Institutes of Health in 1990. They chose a very rare disorder called adenosine deaminase deficiency, ADA, in which the lack of an enzyme disables the immune system, leaving one as defenseless as David Vedder, the boy in the plastic bubble. The experimental subjects were two young girls, 4-year-old Ashanti Da Silva and 9-year-old Cindy